this video, we're going to summarize the two major steps of gene expression. We're going to break down DNA transcription, how we go from DNA to RNA, and translation, looking at how genetic information flows from mRNA to protein. Now, we covered these topics in great detail in the transcription and translation lectures. So in this lecture, we're going to summarize and combine everything we covered in those lectures. So if you haven't seen those two videos, go ahead and watch those first and then come back to this one to review the entire process. Let's get started. Gene expression is where the information encoded in a DNA sequence is converted into either a protein or just RNA. So here we have a DNA molecule, and this right here is the nucleotide sequence of a gene. It's the basic unit of inheritance. It's the segment of DNA that contains instructions for making a particular protein. So when a particular protein is needed by the cell, this segment of DNA is first copied into RNA, or ribonucleic acid, and the RNA copies are then used to direct protein synthesis. So the flow of genetic information in cells is from DNA to RNA to protein, and all cells express their genetic information in this way. This is known as the central dogma of molecular biology, which was named by Francis Crick. Let's now subtract complexity and break down transcription. DNA transcription is quite similar to DNA replication. It begins with unwinding or opening a small section of the DNA double helix to expose the bases on each DNA strand. One of the strands will serve as a template for RNA synthesis. RNA polymerase unwinds the two strands of DNA apart and joins the RNA nucleotides together. Many molecules of RNA polymerase can transcribe the same gene at the same time. And just like DNA polymerases, RNA polymerases can only assemble polynucleotides in the 5' to 3' direction, adding to the 3' end. And it doesn't need a primer to start an RNA chain. So how does RNA polymerase know where to start transcription? Let's go through this. RNA polymerase must be able to recognize the start of a gene so it can bind firmly to the DNA at this position. The DNA sequence where RNA polymerase attaches and initiates transcription is known as the promoter. Now, molecular biologists refers to the direction of transcription as downstream and the other direction as upstream. And these terms are also used to describe the positions of nucleotide sequences within the DNA or RNA. The promoter sequence is said to be upstream from the starting site of transcription, okay? This right here. And the segment of DNA downstream from the promoter, the segment being copied into an RNA molecule, this is called a transcription unit or RNA transcript. Now in bacteria, they have a single type of RNA polymerase that synthesizes mRNA, whereas in eukaryotes, they have at least 3 types of RNA polymerase in their nuclei: RNA polymerase 1, 2, and 3. RNA polymerase 2 is the one used for pre-mRNA synthesis for all protein-coding genes. Now, transcription has 3 stages: initiation, elongation, and termination. Let's go through initiation. To start RNA synthesis, eukaryotic polymerase needs some assistance. It requires a large set of accessory proteins called transcription factors. These proteins assemble on the promoter and help guide the binding of RNA polymerase. Okay, let's break this down further. A transcription factor binds to a short segment composed primarily of T and A nucleotides known as the TATA box. The TATA box is located about 25 nucleotides upstream from the transcription start point. And once it's bound to the TATA box, other factors assemble along with RNA polymerase 2 to form the transcription initiation complex. Okay? And once the RNA polymerase has been positioned correctly, the DNA double helix unwinds, and the polymerase is released from the transcription factors before RNA synthesis can begin. So these transcription factors will dissociate from the DNA, and then they'll be available to start another round of transcription with a new RNA polymerase. So that's initiation. The next stage is elongation. RNA polymerase will move along the DNA, unwinding the double helix and exposing the nucleotides. Polymerase adds nucleotides to the 3' end of the growing molecule. Okay, beautiful. The RNA molecule peels away from the template strand and the DNA double helix reforms with the non-template strand. All right, then the next stage is termination. In eukaryotes, RNA polymerase II is going to transcribe a sequence on the DNA called the polyadenylation signal sequence, this right here, which specifies a polyadenylation signal in the pre-mRNA. So once these 6 RNA nucleotides appear, this right here, they're bound by certain proteins in the nucleus. And then about 10 to 35 nucleotides downstream from the signal, these proteins cut the RNA transcript free from the polymerase enzyme and we release the pre-mRNA. The pre-mRNA will then undergo processing before it can be exported to the cytosol. Recall that translation occurs on ribosomes found in the cytosol. So before it can be exported to the cytosol, it must go through several RNA processing steps, which includes capping, splicing, and polyadenylation. And before these steps are completed, the RNA transcript here is known as pre-mRNA or precursor mRNA. Let's go through RNA processing. During RNA processing, enzymes modify the 2 ends of the pre-mRNA molecule. All right. First is RNA capping, which modifies the 5' end of the RNA transcript. The RNA cap includes a guanine nucleotide with a methyl group, and it attaches to the 5' end. This is the part that is synthesized first. Then the second modification is called polyadenylation. All right. At the 3' end, an enzyme adds 50 to 250 more adenine nucleotides, forming a poly-A tail. These 2 modifications have several key functions. Very important. First, they increase the stability of an mRNA molecule. Second, they facilitate the export from the nucleus to the cytosol and mark the RNA molecule as an mRNA. And third, they help ribosomes attach to the 5' end of the mRNA once it reaches the cytoplasm. Now, capping and polyadenylation occur in all RNA transcripts, but most eukaryotic mRNAs have to undergo an additional processing step called RNA splicing before they become functional. Let's go through this. The sequence of DNA that codes for a polypeptide is not continuous. It's split into segments. Most protein coding genes have their coding sequences interrupted by long non-coding sequences called introns. And the coding sequences are called exons because they are eventually expressed. Think exon to be expressed. Or you can think of exons as sequences of RNA that exit the nucleus, okay? Expressed or exit and express. So introns are removed and the exons are stitched together. Once an RNA transcript has been spliced and both ends have been modified, the RNA is now a functional mRNA molecule that can leave the nucleus and be translated into protein. In the transcription lecture, we covered how the pre-mRNA splicing is carried out, okay? So we've transcribed the gene, modified the ends, and splicing has occurred. What happens now? Only correctly processed mRNAs are exported to the cytosol via nuclear pore complexes. They act as gates that control which molecules can enter or leave the nucleus, and the RNAs that aren't processed remain in the nucleus and are broken down there. All right? That's transcription. The next stage of gene expression is translation. Let's go through this. If you recall in the DNA structure and function lecture, we covered how the structure of DNA carries information for specifying an organism. And we can think of each space, adenine, cytosine, guanine, or thymine, a letter in a 4-letter alphabet that's used to spell out biological messages. DNA and RNA are chemically and structurally similar, which is why DNA can act as a template for RNA synthesis. But when it comes to converting the information from RNA into protein, we're switching languages. We're going to be using different symbols. because there are still only 4 nucleotide bases in mRNA, okay, adenine, cytosine, guanine, and thymine, but there are 20 different types of amino acids in a protein. And so this is where the genetic code comes in. The genetic code is the set of rules by which the nucleotide sequence of a gene is translated into the amino acid sequence of a protein. Let's go through this. The sequence of nucleotides in an RNA molecule is read consecutively in groups of 3. And because RNA is made of 4 different bases, there are 64 possible combinations of 3 nucleotides. As a result, the genetic code is redundant. That is, multiple codons can specify the same amino acid. The genetic instructions for a protein are written in the DNA as a series of non-overlapping 3 nucleotide words. And these mRNA nucleotide triplets are called codons, okay? And each codon specifies 1 amino acid. So during translation, the sequence of codons along an mRNA molecule is translated into a sequence of amino acids making up a polypeptide chain. Codons are written and read in the 5' to 3' direction. So here's the codon table. Here we have the first, second, and third bases. And we have the amino acid in both its 3-letter and 1-letter abbreviations. Now these read here, okay, these are the 3 codons that don't specify any amino acids, but instead they act as termination sites or stop codons, which signal the end of the protein coding sequence in an mRNA. We have UAA, UAG, and UGA. And the way you can easily remember these stop codons is UAA, you are annoying, UAG, you are gone, and UGA, you go away. All right? And there's this one codon here, this green here, AUG, which acts both as an initiation codon and a start signal for ribosomes to begin translation, as well as the codon that specifies the amino acid methionine. Okay, so this is the codon table. Now, codons do not directly recognize the amino acids they specify. Translation depends on adapter molecules that bind to a codon with one part of the adapter and to an amino acid with another. We can call these adapters translators, and they consist of small RNA molecules known as transfer RNAs or tRNAs. So the function of a tRNA is to transfer an amino acid to a growing polypeptide in a ribosome. All right? The cell has all 20 amino acids in the cytoplasm, and the ribosome adds each amino acid to the growing end of a polypeptide. Let's take a closer look at the structure of transfer RNA so you can see how it works, all right? A tRNA molecule consists of a single RNA strand, and its structure can form hydrogen bonds between different regions of the molecule. So it's going to fold back on itself, forming a double helical structure. And there are 2 regions that are very important. At the 3' end here, this region acts as the attachment site for an amino acid. And the other region here, this blue region, forms the anticodon. This loop here is a set of nucleotide triplets that base pairs to a specific mRNA codon. All right? So each tRNA molecule picks up an amino acid in the cytosol, and the cytosol has all 20 amino acids, and it's going to drop it off to a polypeptide chain at the ribosome. And once it leaves the ribosome, it can pick up another of the same amino acid, and the process repeats itself. Recognition of the correct amino acid depends on enzymes called amino-acyl tRNA synthetases, which covalently couple each amino acid to their tRNA. It's very specific. We covered this in detail in the translation lecture. Okay, so that's tRNA. The next important component are ribosomes. Ribosomes are what allow the accurate and rapid translation of mRNA into protein. The structure of a ribosome consists of a large subunit, okay, this bad boy here, and a small unit, each made up of proteins and 1 or more ribosomal RNAs or rRNAs. In both bacteria and eukaryotes, a large and a small subunit join to form a functional ribosome only when attached to an mRNA molecule. So the small ribosomal subunit matches the tRNAs to the codons of the mRNA, while the large subunit catalyzes the formation of the peptide bonds that covalently link the amino acids together into a polypeptide chain. These two subunits come together on an mRNA molecule near its 5' end to start the synthesis of a protein. Gorgeous process. Now, the mRNA has a binding site for the ribosome, which we covered earlier when we talked about the RNA processing. And the ribosome also has a binding site for mRNA, as well as 3 binding sites for tRNA, this right here. Let's break down how the ribosome operates in the process of translation. The ribosome contains 3 binding sites for tRNA molecules called the A site, the P site, and the E site. To add an amino acid to a growing peptide chain, a charged tRNA enters the A site by base pairing with the complementary codon on the mRNA molecule. Consider the A site equals the next amino acid. The A site is also called the amino acyl tRNA binding site. A site, the next amino acid. So the A site holds the tRNA carrying the next amino acid to be added to the chain. Then the P site, or peptidyl tRNA binding site, holds the tRNA carrying the growing polypeptide chain. P site equals polypeptide chain. Then the large ribosomal subunit moves forward, moving the discharged tRNA to the E site before ejecting it. So the discharged tRNAs leave the ribosome from the E site, or exit site. Okay, so let's now take everything we've covered and build a polypeptide. Similar to DNA transcription, we can divide translation into 3 stages: initiation, elongation, and termination. Let's start with initiation. At this stage, we're going to bring together an mRNA, a charged tRNA, remember the first amino acid of the polypeptide, and the 2 subunits of a ribosome. First, a small ribosomal subunit is going to bind to both the mRNA and a specific initiator tRNA, which carries the amino acid methionine. All right? Newly made proteins all have methionines as the first amino acid at their N-terminal end. In eukaryotes, an initiator tRNA carrying methionine is first going to be loaded into the P site for the small ribosomal subunit, along with these proteins here called translation initiation factors. So these factors are required to bring all these components together. The small subunit loaded with the initiator tRNA binds to the 5' end of an mRNA molecule. It binds to the 5' cap, and then it's going to move downstream along the mRNA in the 5' to 3' direction until it reaches the start codon, the AUG. So when AUG is recognized by the initiator tRNA, several of the initiation factors dissociate from the small subunit to make way for the large subunit to bind and complete the entire ribosome. Now, recall that the initiator tRNA is bound to the P site, the peptidyl site. So the available A site is ready for the next amino acyl tRNA, okay? Protein synthesis is ready to begin with the addition of the next charged tRNA to the A site. All right, that's initiation. The next step is elongation of the polypeptide chain. Amino acids are added one by one to the previous amino acid. Each addition involves several proteins called elongation factors and occurs in a 3-step cycle. Let's go through this. The first step is codon recognition. So the anticodon of an incoming amino acyl tRNA base pairs with a complementary mRNA codon in the A site. Then the second step is peptide bond formation. So this step removes the polypeptide from the tRNA in the P site and attaches it to the amino acid on the tRNA in the A site, okay? Then the third step is translocation. What happens is the ribosome translocates the tRNA from the A site to the P site. At the same time, the empty tRNA at the P site is moved to the E site where it is released. Okay? Then the mRNA moves along with its bound tRNAs, bringing the next codon to be translated into the A site. And that's elongation. Now, the next step of translation is termination. So, elongation is going to continue. It's going to continue until a stop codon in the mRNA reaches the A site. Okay, so the nucleotide base triplets UAG, UAA, and UGA act as signals to stop translation. A release factor here, it's a protein shaped like an amino acyl tRNA, is going to bind directly to the stop codon in the A site. The release factor causes the addition of a water molecule instead of an amino acid to the polypeptide chain. And when this happens, this reaction is going to break the bond between the completed polypeptide and the tRNA in the P site, releasing the polypeptide through the exit tunnel. That is gene expression. The genetic information in a cell flows from DNA to RNA to protein. Thank you for watching this video. Make sure you subscribe to EKG Science so you don't miss a single lecture. And remember, subtract complexity and slow down. To study the next lecture, simply click the next video or you can view the entire playlist. Hey, stop procrastinating!